She just woke up, she's healthy. But Allison Landry has been a biology instructor at Louisiana School. She got her bachelor's and PhD in animal science at LSU. She's been at Louisiana School for 7 years. She originally started doing started doing research on cloning as a student worker at LSU. She was in the lab one day. And her husband is a vet. So without further ado, Allison Landry.
I'm not real good at standing still. So to add a little bit to that introduction, and thank you very much for inviting me here today. Both of my parents are Rotarians in the community that I grew up in, and so I've been to quite a few meetings as a guest at various points over the years. And I really appreciate the chance to be here with you today and talk to you today. I had this loaded up, and then my computer went to sleep on me. So I got to LSU after I graduated from high school, and I was really interested in working with animals. And at that point, I actually thought that I was going to be a veterinarian. That's what I thought that I wanted to do at that point. But I got out to LSU, and I had a student worker job that came with my scholarship. This is the days before TOPS. And I thought that I could work in this lab and just get some experience out there. Where I got in this lab was LSU's Embryo Biotechnology Laboratory, and I was fascinated by the research that was going on. They were doing lots of work with cattle and goats. They were doing embryo transfer work. They were doing artificial insemination work, and then they were starting on the cloning research. That started actually after I got to LSU. And while I was there, I worked as a student worker in that lab, and then I had the opportunity to stay as a graduate student. So I stayed to work on my PhD there. And the cloning work was going, and so I stayed in that. And while I was there, I was really interested in all the lab groups, all the groups we had coming through to tour our facilities. We had a lot of high school, middle school students that came through on a regular basis to come through and see our facilities and to talk about our research. And since I was a research associate and a grad student there, I oftentimes had the opportunity to explain what was going on. And so while I was there, I found out that there were a lot of misconceptions about what cloning is actually all about. And so that's why my talk today is on cloning, the science and the science fiction that goes along with it. All right, so let's get going. A lot of people think that cloning began with this famous sheep here. Okay, anybody recognize the sheep? Dolly, all right. So Dolly the sheep. Dolly the sheep was the first cloned animal that was produced. She was produced in England by a group of researchers. And a lot of people think this is where cloning started. But this isn't where cloning started. Cloning had been going on for a little while before this. So if we think about from a natural standpoint, we have natural clones all the time. Identical twins are nature's natural clones. We see those fairly regularly. But from a scientific history of cloning, the first clones that were actually produced by a researcher by the name of Hans Dreich in the late 1800s. He actually took sea urchin embryos, those nice little spiny things. He took embryos and he separated the cells. And what he found was when he separated those separated cells could each form a new individual sea urchin. They were exactly the same, but he was able to separate those cells to create cloned sea urchins. Well, another researcher a few years later tried this out with salamander embryos. And he found these little embryos when they had 2 cells, and what he did was he plucked a hair from his baby's head and took it and tied that hair into a little bitty loop, and then he pulled that loop as tight as he could between those 2 cells and separated that embryo from a 2-celled embryo into 2 single cells. And each one of those single cells developed into a new salamander. So we've got some cloning going on here. Now, when we get into the 1950s, the more modern era of cloning comes around with 2 different sets of researchers, Briggs and King, who were working with frogs, and Gurdon, who was also working with a different type of frog. And what they found was they could produce, with Briggs and King, some tadpoles from those embryos, and then also Gurdon found that he could produce some adult frogs. And now mammals really began to come on the stage after that. Okay? In the 1980s, we first see mammalian cloning. We first see cloned mammals being produced. So what happens is they take an embryo, again, one of these very, very early stage embryos in the 4 to 8 cell stage, and this is an embryo that's approximately 2 to 3 days after it's been fertilized, so a very, very early embryo. You take that embryo and you separate the cells. There's a couple of different ways you can do this, but whatever the method is, you take that embryo and you separate those cells apart, very much like those very early researchers did with sea urchins and with salamanders. And when you separated those cells, you could produce identical offspring. Each one of those cells would become a new individual. And so some of the very early research was done in the 1980s with this method. They would take those embryos, separate the cells, and then they put those new separated embryos into surrogate mothers and let them be grown. And so the first ones of these that were produced were in sheep and cattle. But now it's important to realize this is cloning an embryo. We're not cloning an adult animal here. Okay, we're cloning an embryo. Now, embryonic cell nuclear transfer. You take the nucleus from an embryonic cell. We're not talking about just a whole cell anymore. We take the nucleus from that cell and we put it into an oocyte. This is the female gamete that's had its nucleus removed. And so then you do this procedure, you culture it, you grow it a little while, and then you put it into a surrogate mother. And this first produced mice in the 1980s, and followed by that was sheep and then other species, cattle, etc. And then in 1996, this is the first time that we see cloning of mammalian embryos from a cell that started to acquire a different function. This is from a much older embryo. This is from an embryo that's much older than 2 or 3 days. This is from an embryo that's over a week old. And at this point, this embryo is starting to form separate tissues. An embryo over a week old is starting to form some nervous tissue. It's starting to separate out into those different tissue layers. And they found that if they took some of those differentiating cells that were starting to acquire some different function, they still could actually clone that embryo and produce a new offspring. Interestingly enough, that study in 1996 is the same group of researchers that produced Dolly a year later. So this is kind of their stepping up towards that policy. So somatic cell nuclear transfer. This is what we technically call the cloning process that produced Dolly in 1997, SCNT. Dolly is so important because she is the first clone of an adult mammal. She is the first clone of an adult mammal. There have been lots of mammal embryos that have been cloned before that, but never an adult mammal. And so that's why Dolly made such a big splash. She was the first adult mammal to ever be cloned. Dolly is no longer here on Earth today. She died when she was somewhere between 8 and 11 years old. I don't remember the exact date. She's passed away now, but she was the first one to be cloned. It wasn't long after that that numerous other species were cloned. At this point, there have been quite a few species: dogs and cats, horses, a white-tailed deer, cattle, various exotic species. One of them being an exotic cattle species called the gaur. There have been quite a few other successes in other species since Dolly. Now, what I want to do in the next little bit of my talk is talk to you about the science of cloning and the steps that go into cloning an animal. I'm going to show a few video clips from when I was actually doing some cloning procedures. Now, the steps I'm going to show you are actually going to be cattle. So please don't get real excited. We're not cloning people or anything here. This is video from me looking at cloning a cow. So I'll show you some video clips for actually going through the process. But some of the steps, and you can see here, we'll go through each of these steps as we move along. Okay, so our first step that we have to do is we have to identify the superior animal that we want to clone. Now, this may be a cow that produces the most milk that anyone has ever seen. This may be a steer that cuts out at the best amount. It has the best fat to lean ratio, it rates choice or prime, whatever that is. But in some case, whatever it may be, you identify your superior animal. This could be your favorite dog that you want to clone. And yes, there are companies out there that will clone your dog for you if you want to do that. Or it could be the championship racing mule. And I don't know if you knew that there were championship racing mules, but the first mule that was ever cloned was the champion racing mule. So when we think about this, what we do is we take a skin cell, and that little cartoon up there was my attempt to make a skin sample in PowerPoint. But when you see that little skin sample, what you do is you take a biopsy. You just take a little biopsy punch and you take a skin sample. You can also clone animals from other tissues besides skin tissues. But you take your set of cells and you grow them on a petri dish in the appropriate environment for them. So this has all the nutrients they need. It's grown under the appropriate oxygen levels and everything else. And you get those cells growing nicely in your incubator. This is an image of what some of these cells from a skin sample would actually look like when they're growing under an incubator. You can kind of see here, let me see if I got it for it. That long little stretch right there is actually one single cell. And so there are quite a few cells in that image on that screen at this point. And so you get this selection of cells growing, and you get your cells growing. And in order to get those cells ready to clone, you actually have to treat them a little bit chemically. First, you actually starve them. You take away some of their nutrients, and it sends them into this stage that makes them more suitable to be cloned. Now, the next stage is to take your oocyte. So you've got your oocyte. And the oocyte, this is our female gamete here, our oocyte has a nucleus, and so you see that little circle right there at the top, that's to indicate where the nucleus is in this oocyte. Now, mammalian oocytes aren't transparent. You can't just look at an oocyte and tell exactly where that nucleus is. And so in order to see this, a lot of times we would actually stain our embryos with a fluorescent dye and then look at them under ultraviolet light so we could see where that nucleus would be. And so what we're going to do is take right there this glass pipette, and we're going to use that glass pipette and basically a hydraulic setup to apply gentle suction to pull out that nucleus. And so this is going to be a video clip that shows that process of taking out the nucleus of an oocyte. So there's my oocyte on the screen. There's my ultraviolet light. You can't really see the nucleus at this point because the gain's up really high. But there's my pipette on the side, and so you'll see it puncture in. And I'm going to apply a little bit of suction, and you'll see that begin to pull back out. Do you see how bright it is when that pulled out? That's the nucleus. Okay? And so I've got that material in my pipette, and I'm actually going to, I'm going to check it to make sure I've got all of that genetic material out. And then I'm just going to get rid of that. And so that's the end of that one clip right there. Okay, now once we get rid of that piece of genetic material inside the oocyte, our oocyte is ready to have the new cell that we want to clone with the nucleus, the DNA from that animal we want to clone, put back in. So I have my enucleated oocyte. Here's my oocyte. You can see there's no nucleus in there. And this is the cell. That's the cell that I want to clone that's up there at the top. I do have more room? Oh, okay, thank you. Okay, so this is my cell this is my oocyte without the nucleus. This is my cell that I'm bringing in. This is my pipette bringing with the little arrow showing it being put in. This is the cell I've been culturing, and there's its nucleus right there. And so that's what my second video clip is going to be, is actually showing you the process of putting that cell into the oocyte that's been enucleated. Okay, so this is actually later the same day. Okay, I'm going out of focus here of the oocyte so you can see those cells. Okay, and those cells are now kind of rounded up as we've taken them off the bottom of the petri dish. I'm going to draw it in, and it went away. I had too much suction on there. It'll come back. You'll see it in just a second. You'll see it in just a second. You'll see it kind of come in from this side. There it is, right there. Okay. So I'm going to bring this back in focus, and I'm going to bring my pipette back into focus. Yeah, a micromanipulator. Very good. All right, and so what we're going to do is roll the oocyte to get to the point where I think I've got the best chance of putting in that particular cell. And so I'm going to get everything adjusted. Oh, I'm losing my cell. So I'm going to draw back in. Okay. And so you can see I'm just kind of nudging that oocyte to get it moved around so it'll be in the best spot, and where I have the best contact between the cell membrane of the oocyte. And so what I'm going to do is draw in a little bit of that oocyte and then push my cell in. And why I do that is to get good contact. And so I'm going to get that in there. I'm going to kind of tuck him in just a little bit. So I maintain that good